Okay. Good morning. So thank you to the few of you who are here for being here. I know a lot of people are already traveling for the holiday weekend. So for those of you who are listening via the podcast, happy holidays to you. That's not you, but maybe you're out there on the internet somewhere. So happy Thanksgiving. I think it's always famous last words, excuse me, for me to say this, but I think that I should be able to finish in under time today. Highly unusual, so no promises. I have a tendency to talk a lot. But I'll do my best. I was reviewing my notes this morning, and I think we've made good progress in our discussions of the mechanism of translation, and that's what we're largely going to finish today, and then talk a little bit about the topics of what happens to certain proteins after they are translated by the ribosome, and different trafficking pathways, and just sort of in a cursory manner touch upon some of these post-translational, we would say, modifications and trafficking pathways. But before we do, I thought we would talk a little bit of holiday biochemistry. Seems appropriate. So we all have heard the statement, right, that eating turkey makes you tired, right? Why does eating turkey presumably make you tired? Does anyone have any thoughts about that? I mean, the answer may or may not be on the board. What's that? I'm hearing rumblings. It's tryptophan, right? Is that what you think? Right? So there's a pathway. It's certainly true that when you eat turkey, for those of you who eat turkey, that there is, as we've learned, there's a lot of protein in muscle tissue, meat, right, like turkey. And there may not be a lot, but there's certainly some tryptophan, the amino acid, that you guys are acquainted with, as a component of protein in turkey, right? And it is also certainly true that what I'm showing you here, which, mind you, for those of you who may or may not be concerned about this, will not be on the exam, okay? But this is a pathway, right, that discusses or depicts the biosynthetic pathway of several molecules you may have heard of. For example, serotonin, which is a molecule that is able to help one relax and essentially get into a sleep state. And also, that's on pathway to melatonin, which you may have heard of as a compound that some people actually take in order to combat the effects of jet lag when they travel, right, to help them sleep better. So it is certainly true that tryptophan, which is a precursor in the biosynthetic pathway of both serotonin and melatonin, is consistent with the hypothesis that eating turkey makes you tired, okay? So I could say, well, wow, you guys should memorize this pathway and go home to your families or whoever you might be celebrating the holiday with, if you are so inclined, and school them, right, by drawing out this complex biosynthetic pathway and say, look, you know, this is how tryptophan is making you tired. However, there's an assumption that goes into this hypothesis that eating turkey makes you tired, right, as compared to, say, other meals. And what is that assumption? Or that turkey is somehow unusually rich in tryptophan, right? So would it be different if you had steak, right, for Thanksgiving dinner, for example? The answer is no. It would be no different, okay? So turkey is no more abundant in tryptophan than any other sources of protein that we tend to ingest, whether they be animal products or otherwise. And so it's actually a myth. So the answer to this question is no. Eating turkey in and of itself does not make you any more tired than just eating a lot and probably drinking together with your eating, which may or may, again, be something that you do. May not, depending on your personal preferences. I will be, okay? And I'll be tired, right? But I'll know that it's not the tryptophan in the turkey that's made me tired. I'll know that it was the third helping of turkey and stuffing and whatever vegetable I might eat, together with the wine and the beer I might ingest, okay? That that's what's making me tired and not the turkey, okay? So if you want to school your friends, families, relatives, what you really want to tell them is that this is a myth, okay? Many people talk about this, but you've learned now in biochemistry that tryptophan is in fact a very rare amino acid. You might want to just bust out the structure of tryptophan for them. That could be impressive, right? But nevertheless, you should tell them that it's not eating turkey that makes you tired. It's just eating, right? That makes you tired, okay? So we open with that, and that's all I have to say about that. And plus, I like this dancing turkey. Okay. Okay. So coming back to the topic of the day. So I thought we would just backtrack. We covered a lot of sort of detailed mechanistic information about how the bacterial ribosome initiates the translation process. I'm not going to go in step by step, but suffice it to say you have these initiation factors, or these IF proteins that are required to set up this pre-initiation complex. In the process of doing so, a GTP molecule is bound and hydrolyzed, specifically by the IF2 cofactor, which helps to stabilize and deliver that special tRNA FMET initiator molecule. And once that pre-initiation complex with the small subunit of the ribosome is established, that serves as a signal for the large subunit to stably associate with the rest of the complex in order to establish what we then would refer to as the initiation complex. Okay? So that's all by way of review. We talked a little bit about this idea of specialized sequences in messenger RNAs that help the ribosome through interactions, here shown as the depiction of a little tail of the 16S. That's one of these large RNA molecules. It's the RNA molecule that constitutes structurally, together with its protein counterparts, the small subunit of the E. coli ribosome. And through interactions with the 3' end of that RNA and this specialized sequence that we call the Shine-Dalgarno sequence, which again I wanted to place into the context of other things that we've discussed in the class, right? Like promoter sequence elements, helps to orient the machinery of translation in such a way so that this start codon here is properly positioned within the complex, which when I say properly positioned, I mean sitting here right at the small subunit interface of the so-called P site, or the peptidyl transfer site. So I'll just also remind you that in the ribosome, and when we talk about the process of translation, we want to be thinking about these 3 sites that can each individually accommodate a tRNA together with its covalently coupled amino acid. The A site, or the amino acid site, the P site, or the peptidyl transfer site, and this E site as the exit site. So just to remind you again, the process of translation involves a movement of tRNAs, right, through this protein synthesis machinery, the ribosome. Okay? And so I think we got to this point, which is that very first elongation step. So where we're now transitioning from the formation of a stable initiation complex to the formation of that first peptide bond. Okay, so this would be going from individual amino acids to that first dipeptide that we're going to form in the early stages of polypeptide synthesis. And that involves yet another factor that I pointed out called elongation factor TU. And that's an important one because that's effectively a delivery protein that binds to the charged tRNA. So this is the tRNA carrying its covalently associated amino acid off of its very 3' CCA terminal end, right? We recall all of that stuff. And that EF TU factor, together again with its GTP bound in its nucleotide binding site, effectively helps determine whether the appropriate tRNA is binding in the A site, okay? And that is essentially, I used the term sampling the last time, but it's essentially querying or sampling that codon-anticodon interaction. And when the appropriate codon-anticodon interaction is formed, that makes a more stable complex of the EFTU tRNA amino acid complex in this A site. And that increased stability signals to EFTU that this is, in fact, the correct tRNA to be delivered at this point. And that signal itself is transmitted or enacted through the hydrolysis of the GTP cofactor. So basically, that GTP cofactor will not be stimulated to be hydrolyzed until a stable complex in the A site is formed, right? So that's essentially a way of making sure, in a sense, it might be a little confusing, I imagine, to you, because I talk about how the ribosome doesn't really do fidelity checking, right, at the level of the amino acid, right? We talked a lot about how the amino acyl tRNA synthetase charging enzymes that put the amino acids onto the tRNAs are very important for maintaining the fidelity of the translational process, which is true. But now I'm telling you that there is some sampling and checking going on in the ribosome. But I'll differentiate that. So it does have fidelity checking with respect to the appropriate tRNA, but it doesn't necessarily know what kind of amino acid has been coupled here. So that's the distinction, right? So it's making sure that the cognate tRNA is the one that is going to be bound. So that's the decoding step, in essence, by the ribosome, okay? And then a GTP is hydrolyzed. That releases this EFTU elongation factor. There is this recycling pathway to free up EFTU to bind to another GTP precursor so that it can bind to another tRNA and repeat its function. But we left off here with the product of that recognition event, right, where you have now deposited the tRNA in the A site. You have your initiator tRNA that was part of the initiation complex assembly pathway positioned neatly in the P site. And now chemistry is poised to happen, at least as it relates to peptidyltransferase in the active site of the ribosome. So that's where we left off. And so now we can zoom in and have a look at the peptidyltransferase reaction. And what you can see here, so we're just zooming in now into the active site. You're not seeing it, but there are actually RNA nucleotides that are helping to mediate the chemistry that is being drawn out here, but it's removed for simplicity. And what you can see is that here we have the linkage between the, let's see here, between the FMET and this carboxyl terminus, and then you have this amino terminus of the incoming amino acid. And essentially what the active site of the ribosome does is activate this amino group to serve as an electrophile, I'm sorry, a nucleophile to attack this carbonyl group, okay, which is the electrophile. And then, again, this is just another example of one of these inline attack reactions that we've seen so often here. And so in this case, you get this transient intermediate transition state, where in the transition state, you're sort of both covalently coupled to the P site tRNA and now the amino acid on the incoming A site tRNA. But eventually, that transition state relaxes down, which essentially results in the formation of the first peptide bond, which is drawn here, right, between now what was that attacking group, that amine, and the carbonyl group on the preceding amino acid. So at this point, what you've done is you've taken that FMET and you've transferred it to the tRNA that is residing in the A site, okay? And you've now removed any amino acid from the tRNA, which is in the, well, it's already been shown to move here, but essentially this chemistry is happening such that transiently you have the A site have the dipeptide, and then the P site becomes vacant, and then very quickly, in a step that they're skipping over here, what ends up happening is that the ribosome then begins to move, okay? And it transitions into what is referred to as the hybrid state. I'll describe now what that means of the ribosome. It's referred to as the hybrid state because if you look here, let's look at the zoomed out version for a moment. Notice that the anticodon stem loop end of the tRNA has not changed its position with respect to the ribosome. So the A site occupying tRNA, at least with respect to the anticodon stem loop, is still in the A site, right? The P site initiator tRNA, together with the start, cognate start codon, are positioned in the P site. The E site down here on the small subunit is also vacant. Okay. However, so that would suggest that no movement has occurred along the mRNA yet. However, if you look up at the large subunit, concomitant with the peptidyl transfer reaction is the beginning of what we would refer to as the translocation step of the ribosome, which is needing to move down this mRNA as it decodes codon after codon after codon. So an intermediate in that translocation process is essentially referred to as this hybrid state where the acceptor stem, where the amino acid is initially coupled, those parts of the tRNA move into the next adjacent site. So in other words, that acceptor stem, which was here in the P site, once the reaction is complete, moves over into the E site, okay? And the acceptor stem, which is receiving essentially that dipeptide, which initially began in the A site, upon completion of chemistry here, finds itself in the P site, okay? So the way I like to think of this is that the tRNAs during chemistry sort of tilt over, okay, on the acceptor stem side of the tRNA, but the bottom part, if you will, to use, using this frame of reference, this lower part of the complex has not moved yet, okay? And so because you have sort of this partially translocated state, that's why we refer to this as the hybrid state of the ribosome, because half of it is sort of translocated with respect to the large subunit, and half of it is not. Does that make sense to everybody? Okay. So that is the concept of the hybrid state. So let me just define that a little bit more. So during peptide bond formation, the acceptor stems move into, let's just say, adjacent sites. But the anticodon stems do not. And that's what gives rise to this hybrid state. Okay? Are there any questions about that? Okay, so that's an important intermediate. And now the next question, right, that one should be thinking is, okay, well, now you've done the chemistry required to form that peptide bond. So, but the machine now is kind of clogged, right, because you need to now vacate that A site so that the next tRNA can come in. And the other thing you need to achieve is you need to move the next codon, which would be down here. In this drawing, there's going to be another codon right here, right, that needs to get into that A site to be decoded, okay? And that step where everything moves, right, with respect to the frame, the open reading frame of the tRNA, that's what we refer to technically as the translocation step of the ribosome, okay? So that's what we're going to talk about next, okay? So during elongation, we're going to discuss how is it that the ribosome translocates along the messenger RNA. Okay, so here's another cartoon which picks up from where the last cartoon just left off. This is now, you can see the hybrid state. So let me just write that of the ribosome. And now we have a second elongation factor. So far I've discussed with you elongation factor TU, helps to deliver the tRNA to the A site. Now we have another really important elongation factor, which is called elongation factor G. Okay? And this is a protein elongation factor, as is EFTU, which is essentially the translocation factor. Okay? So how does that work? So basically, the EFG protein can come in. So once this complex is sitting here in the hybrid state, that then allows the EFG protein to be accommodated such that in steps that are, the mechanism is definitely not entirely understood at this point, the presence of this EFG, again, notice there's another bit of energy being committed here, plus a GTP cofactor, binding near the A site helps through the energy of GTP hydrolysis for the entire complex to translocate. So essentially, EFG bound to GTP binds. Okay, I think from here to here, there's effectively no, this is the same complex, right? And then remember, what we're trying to do here is push this machine along. And so effectively, what will eventually happen is that EFG will essentially, through the energy of GTP hydrolysis, force its way to occupy that A site, which then pushes the tRNA that's down here in the A site in the small subunit into the P site, and also the P site tRNA into the E site. And that is what you arrive at here in the fully translocated ribosome complex, right? So really, this step right here is the translocation step. And notice the result of that translocation then becomes the used up tRNA that no longer is charged residing in the E site. The tRNA that had previously been delivered to the A site now residing in the P site, right? Together with the growing peptide chain, okay? And now that leaves this protein EFG GDP in the A site, which in the GDP bound state has a lower affinity for the A site and will intrinsically dissociate at that point, okay? And so that's essentially getting you ready for the next tRNA. So this will eventually dissociate, okay, to allow another round of translocation to occur. And then this process will repeat. And so essentially, you'll have GDP fall out here, or dissociate is a better term, leaving this site vacant. And I'm just going to flip back for a moment. And then once that site is vacant, you're essentially back to, I should also point out that the affinity of the E site for tRNA is quite low. So once a tRNA gets into that E site, it just diffuses away to be recycled. So essentially, following this process, right, this complex very quickly gets to look something like this complex, but you just have 2 amino acids, right? And the process repeats. Is that making sense to some of you? Most of you? All of you? Okay, that's good. Okay. So there's something interesting, I think, about EFG GDP, which is already sort of implicit in this cartoon. So before we being the collective we of molecular biologists and biochemists who are interested in structure-function relationships of molecular machines, like we're discussing right here, it had long been known that the EFG factor was very important for that elongation step. And if you have mutations or you're missing this factor, the ribosome does not effectively elongate and translocate. But it wasn't so clear what at least the beginnings of the molecular basis for how EFG actually achieves this stimulation of translocation until the crystal structure, meaning the atomic resolution structure of EFG, was solved. And what's really interesting to think about is how evolution has given rise to an elongation factor. So here's EFG, right? All protein. This is just a protein cofactor of translation. And notice that its shape, right, and size are remarkably similar to the EFTU protein bound to a tRNA. So this is a protein RNA complex, right? And so notice how overall the shape, even to the level of ridges, so this is protein here, this is a tRNA helix here with its phosphate backbone. So notice how the protein has evolved to take on the shape of a tRNA bound to the EFTU factor. Isn't that cool? That's interesting, right? And so we call that structural mimicry, right? And that begins to inform our understanding of how EFG works, right? Because the site where it needs to bind in this figure is interested in accommodating a molecule of a particular shape, right? And so as EFG evolved to become this translocation cofactor, there was a selective pressure for it to take on a structure that looked very similar to the tRNA bound with an EFTU cofactor, okay? And that didn't necessarily have to be the case, but that's how it works, okay? So this is an example of this concept of structural mimicry, and that does come up with some frequency in biochemical mechanisms where a molecule that needs to compete for a binding site with another molecule, as you might expect, even if it's not made of the same substance, in one case RNA, in the other case protein, can take on structure that mimic each other, okay, and thereby compete, okay? So I just thought I would point that out, okay? So that cycle continues, right, depending on how long the protein that's being encoded is, right? And as we know from our discussion of the genetic code, the ribosome will just cruise along at a pretty solid clip and move along the mRNA until it reaches one of these three stop codons that we talked about, right? So the stop codons that you can look up on the genetic code table are UA, UAG, and UGA, right? And remember, these stop codons, there are no tRNAs that recognize them through their anticodon stem loop interaction. And so essentially, once you get excuse me, to one of these stop codons, which is being shown here, actually, this gives me an opportunity to point out another feature of the ribosome, which is important, which is noticed here. So the growing polypeptide chain, right, is exiting. It's not unlike when we talked about transcription and there's an exit tunnel for the nascent RNA. There's also an exit tunnel from this machine for the nascent polypeptide, okay? And so as more and more amino acid is being linearly added and decoded from the mRNA, the polypeptide is exiting this exit channel. So this is the polypeptide, let's say, going through the so-called exit channel. So that's its way out to the cytosolic world, right? And some interesting things immediately begin to happen, as we'll come to in a moment, as proteins emerge from the ribosome. There's some interesting questions about folding of the protein, right? We've talked a lot about structural properties of proteins, how they have to fold. Well, now we have to place that into the context of, well, how does folding get impacted by the fact that part of the protein is extruded, right, before the rest of it is even made, right? And those are interesting questions that do not have necessarily simple answers, but it's still interesting to think about. But let's, again, focus in on what happens at the end. So this is now the final act of translation at the ribosome, where you reach one of these stop codons. There's no tRNA, but rather, again, in keeping with the same sort of theme, there is a protein cofactor called a release factor, or an RF, release factor. Okay, and this is a protein cofactor, which also resembles the shape of a tRNA, but can, interestingly, so it binds to the A site, and through protein RNA interactions, it can actually recognize one of these three codons that are stop codons. So there's bits of the RF protein, that is polypeptide. There are different release factors for the different stop codons. And they specifically recognize UUA or UAG or UGA. I think it's actually not as simple as a one-to-one. I think there are 2 release factors, and one of them can recognize 2 of the stop codons. But suffice it to say, there's protein RNA specificity. So it binds to A site, and then the protein specifically recognizes the stop codon. And the presence of and again, this is something that I think, I know that, for example, people like Harry Noller and his group members are very actively pursuing right now, is trying to understand how it is that accommodating a release factor, okay, how you get from this intact ribosome, which remember is being grossly oversimplified here, right, how you go from that release factor to essentially the disassembly of a ribosome into its components so that they can be recycled. So there are major questions structurally about how the accommodation of a release factor triggers you see here, EFG-dependent recycling of all these factors. And this just sort of scratches the surface of what happens. This now begins to go back to that very early slide of the formation of another initiation complex, where one of those early steps is binding of IF3, that initiation factor, to the E site, right? You might recall. So you can see that that sort of connects the loop, that an aspect of the termination process here is actually also a facet of the initiation process for the ribosome to then be recycled onto another mRNA, which sort of makes sense. But again, the details here are not completely worked out. But the key point here is that there is a protein release factor through recognition to a stop codon. Let's just say it triggers through some mechanism the disassembly of the ribosome and recycling. Okay, it's important to note that these parts, right, of the cell is not interested in just throwing these things away with each round of translation, but rather recycling them to continue the decoding process on other messenger RNAs. Okay, so in summary, what we've done here for the bacterial system is we've gone into some detail about how the initiation complex is formed and how IF, or initiation factors, are required. Then we talked about the elongation process and how there's requirement for elongation factor TU and elongation factor G to help deliver the appropriate tRNA to the appropriate ribosome site, EFG helping the whole complex move along once the peptide bond has been synthesized. And then once many cycles of those steps have passed, the elongation steps have been reiterated, then eventually you find yourself at a termination codon, and that, as its name would suggest, terminates the process, again with the help of protein cofactors, in this case, a release factor. You have a question? UAA. So no. So there are 3 different stop. So this says UAG, yeah? So there are actually 3 different stop codons. Let me show you that. Dancing turkey. Right. Sorry. Here. So let's look at that. So here's our genetic code lookup table. So let's look at the stops here. UAA is a stop codon. So your question is, should it be UAA? It could be UAA, but I think that the figure is showing UAG. That's also a stop codon that is recognized. Is that correct?
I think she's saying he wrote 3 stop codons and he wrote UAA.
Oh, I did? Is that what you were saying? Damn it. So I just miswrote it. Sorry. Here. UAA. That's what you're asking, right? Sorry. Yeah. My bad. Thanks. Okay. Thanks for pointing that out. Okay. Good. So a few of you who have come to office hours throughout the quarter, we've talked about this concept, and I think that this relates to a bunch of different questions that have come up about, what is moving with, there have been questions as we've talked about these molecular motors moving along their tracks, asking about what is moving with respect to what, right? And is it a matter of sort of reference point, perspective, or is there something more to thinking about that? And in the case of the E. coli or bacterial translation in general, unlike, as we've discussed in some length, unlike in eukaryotic cells, where the processes of transcription and translation are spatially separated from one another, right? Where in a eukaryotic cell, transcription is occurring inside the nucleus, right? And that mRNA, after all the processing steps that we've discussed, gets exported through the nuclear pore, which I'm going to show you a picture of in a moment, and then translated in the cytosol. In bacteria, everything is co-localized in a sense, right? Occupying the same space. And that gives rise to this idea of co-transcriptional, or phenomenon, of co-transcriptional translation, okay? So the idea of co-transcriptional translation really dates back to early EM images. So let me just define what that means, right? So here we have essentially the two latter steps of the central dogma wrapped up in one diagram, right? Where basically you have the DNA, an RNA polymerase, which is actively elongating and synthesizing RNA. It's exiting out the exit channel. And then ribosomes can immediately begin to assemble on the 5' end of that RNA. And actually, so here's one that's just coming in and starting to assemble. These guys have already assembled and are chugging along making protein. Okay? And so you have this real assembly line of protein synthesis where literally as the RNA is emerging from the RNA polymerase machinery, it's immediately engaged by the ribosome and being made into protein. Okay? Now, the notion that co-transcriptional translation may or may not occur, so there's this term called polysomes, which is simply, as its name might suggest to you, the idea that multiple ribosomes can together be associated on a single messenger RNA, okay? And that's true for eukaryotic RNAs as well in the cytosol. There'll be many, depending on how strongly expressed a particular protein is. There'll be many ribosomes on a single mRNA, which gives you a sense of the amplification that can occur during the protein expression process. From one mRNA, you get many proteins, okay, being synthesized in sequence here. But I think that this is kind of neat. This is an old EM image where this idea first arose, where you could see, literally, if you prepared DNA from metabolically active bacterial cells carefully enough so as to be able to open up and gain access to the contents of the cell without completely destroying the structural properties, which is not a trivial thing to do, you can identify that there's a piece of DNA, which is false colored in this blue here, and then the site of the promoter for the RNA transcriptional process in the DNA is located by this arrow, right? So this is the promoter site. And notice that so essentially what you should think of here is that although this is a snapshot image, this is essentially the progression of many RNA polymerases moving along the DNA in time, right? You can think of it that way. Such that as you move linearly from left to right along the DNA, as you would expect, more RNA has been transcribed out of each of the individual RNA polymerase complexes. And as a result, you see more and more of these very electron-dense spots, which are the ribosomes, okay, in the EM. And notice that they get longer and longer the further you get from the promoter, okay? So this idea of co-transcriptional translation and the concept of polysomes really emerges from this single image, right, where you can correlate how many ribosomes and the length of the RNA with how far you are from the promoter, That's pretty wild, right? That all of this can be happening in sort of a connected fashion, right? Everything happening very fast and coordinated. And you might say, oh, well, co-transcriptional translation, why does that happen? Why is that an important feature of prokaryotic gene expression and has been lost, in essence, in eukaryotic gene expression because we've decoupled these two processes? And one rationalization that you could come up with, or that I could come up with, is timing, right? So, everything happens very quickly for a bacterial cell. You're dividing every half hour or so, right? And so in order to make enough protein for the daughter cells to continue to survive, you sort of need these processes to be coupled to one another in time so that everything can happen fast, right? Whereas there's not nearly as much selective pressure on a eukaryotic cell, which might divide every 12 to 24 hours, right, to sort of get it all done quickly. As in contrast, there's a lot of pressure to make sure that you get it done right, okay, in that case. Whereas a bacterial cell in an exponentially growing population, there's sort of this trade-off. Some cells can mess up. In fact, from an evolutionary point of view, it's somewhat advantageous for them to mess up a little bit because as a population, if the environment suddenly changed, some of those messed up or deviated E. coli cells might somehow gain a selective advantage, and there's sort of interesting arguments to be thought about in that context. Okay, but I think it's really useful to, and the reason why I stick on this slide for a while is because of the slightly annoying way that we're forced to teach biochemistry, right? Where we have to segment each of these processes into its corral, into its own chapter, right? And there's an argument, pedagogically to do that so that we don't just, completely bowl you over. But at the same time, it's nice to wrap it back once you've digested some of that material and realize that it's all happening, right, at the same time, right? Which is pretty exciting, I think. Okay, so that's protein translation. And in the few remaining slides that I have to show you today, I just want to talk a little bit about what can happen to proteins after they're synthesized by the ribosome. Okay? And some of these things we've already talked about, so we can go through them fairly quickly. So for example, we talked about the idea of post-translational modification. We've also talked about post-transcriptional modifications of RNA. So once these biomolecules are made, there's further modification. We talked a lot about post-transcriptional modifications in the context of, say, tRNA, structure, function, recognition by tRNA synthetase enzymes, so on and so forth. Now we're talking about post-translational modifications. What can covalently sort of alter the structural properties of proteins after they've been made? Many of these post-translational modifications serve to modulate the functional state of a protein. Literally can be thought of in certain instances, particularly with respect to, say, modifications like phosphorylation, the addition of a charged phosphate group to either a serine moiety, so the alcohol-bearing amino acid side chains, serine, threonine, and tyrosine can all be phosphorylated by this class of enzymes that I mentioned in passing called kinases, right? So you have enzymes called kinases, and they can catalyze this phosphorylation reaction, which is the addition of a phosphate group to a solvent-exposed serine, threonine, or tyrosine. And this phosphorylation event turns out to be an exceedingly important modulator of many protein-protein interactions. Okay? So in the cell, if it's not already immediately obvious from the discussions we've had, for the cell to function, proteins must come together in a regulated fashion and stably dissociate at the appropriate times. So one of the ways that the cell has evolved to control when proteins stably dock or are made to be less stable is through the introduction of phosphate groups on certain surfaces, which can be binding interfaces for protein-protein interactions. So you can think of phosphorylation as sort of a binary code that decorates or changes the chemical properties of a protein to say when it's time to undergo a functional interaction or not, right? And so there's a lot of really interesting discussion to be had about the role of phosphorylation and dephosphorylation. So kinases add a phosphate group, a class of enzyme called phosphatases. Those enzymes remove phosphates and often do so in a very sequence-specific manner. So there'll be a particular string of amino acids within which, say, a tyrosine might exist that is recognized by a specific kinase at a specific time, say, in the cell cycle, by way of example. So that's one type of very important and universally observed post-translational modification. Another one that I'll point out is methylation, particularly on, let's focus on the lysines. So we talked about, so lysine methylation, we talked quite a bit about nucleosome structure, the core nucleosome particle and its histone proteins with the histone tails that are flapping in the breeze that are very lysine rich. Okay, I'm just reminding you of some of these details. And those lysines can be methylated, and those methyl marks on nucleosomes can evoke certain, let's say, activity states of particular regions of the genome. Okay, so you can have very high levels of methylation correlating with low gene expression states, or otherwise silencing is sort of the term that's used, whereas removal or demethylation can sort of serve to activate regions of the genome. Okay, and when we think about these methyl marks on nucleosomes, right, that's sort of a level of genetic control that is sort of one level above what we're used to thinking, which is the DNA level. And so the term that we use when we talk about these, this is just one example of a type of modification that alters the genetic regulation of the genome, we refer to that as epigenetic marks or epigenetic regulation. Okay, so I'll just introduce that, genetic marks. Okay, so these are just a few very arbitrarily selected but very abundant post-translational modifications that take sort of the functional properties of a protein and add an additional layer of regulation through addition and removal of certain functional groups like methyl groups or phosphate groups. Okay, so we'll just leave it at that for the moment. Okay, so somebody asked me a question when I showed the picture of the rough ER, right? And we talked about how what makes the rough ER rough is the ribosomes that are stably associated with the endoplasmic reticulum membrane. And so I think after that lecture, one of you guys came up and asked, well, how does a ribosome know to associate with the ER? So that's what's being discussed here. So how is it that certain proteins are targeted to certain regions of the cell? Okay, and this is a very complex topic, protein targeting. You could imagine, especially in the context of a sophisticated eukaryotic cell, it's non-trivial, to say the least, right? But we can start to think about, and it's fundamentally critical, right? Because in order for a protein to exert its necessary function, a big part of that comes down to where it is, right? Where it's so-called localized inside the cell. So if a cell, for example, or a protein, for example, needs to exist on the outer surface of a cell, say in the context of a receptor protein that needs to bind to some extracellular molecule to sense its environment, how does that receptor protein get to the outer surface of the cell? That's a question, right? Or if there are proteins that need to actually be secreted from a cell, right, to then diffuse away from the cell and communicate with nearby cell within the context of a tissue, right? So how does a secreted protein know, right, or ultimately find its way to be secreted, okay? Well, a lot of that relates to what happens in a co-translational manner. And so let me just walk you through this schematic here, where this is all stuff we've already talked about, where you have the assembly of an initiation complex. It's drawn right to left here. AUG is the start codon. Okay, so we're moving in this direction. Okay, and so it's assembled here. It begins to move along. The protein emerges from the exit tunnel, and there, for some subset of proteins, there is a consensus sequence on the very N-terminal side of the protein, which is referred to as a signal sequence. And for this class of proteins, that signal sequence serves as a recognition motif for a complex, which happens to be a protein RNA complex, which is drawn here as this little green cylindrical thing. That SRP stands for signal recognition particle. Okay. So as the signal sequence emerges from the ribosome, the signal recognition particle can engage that nascent polypeptide as it's actively being translated. And this is all happening in the cytosol. But as soon as that signal recognition particle is bound to the signal peptide in the newly synthesized protein, that complex actually then results in a stable association of the entire translating ribosome together with its newly formed peptide onto a pore or a translocation complex that exists on the endoplasmic reticulum membrane. Okay? So this is a very sophisticated system where you have essentially those ribosomes which are making proteins that need to be translocated as part of their maturation pathway into the inner so-called lumen, the inner space of the endoplasmic reticulum. That all happens co-translationally. Okay? Again, this is a simplified cartoon. The exact way in which this interaction occurs, signal peptide is sort of shunted through this protein translocated machinery. There are some major open mechanistic questions there. But from a phenomenological point of view, we know that it occurs. And the important thing is that proteins with this signal peptide are then ultimately, upon completion of translocation, are essentially pushed in or translocated into the ER lumen. I should have said upon completion of translation, that protein is translocated into the ER lumen. And then, as I'll show you in a moment, being in that ER lumen space is the starting point for a lot of modifications and trafficking pathways that either get proteins out of the cell, secreted, or get them displayed on the surface of the outer membrane through a variety of pathways. Okay? So key points here is that for proteins that need to be secreted or displayed on the surface, they bear a specialized sequence. Interesting experiments have been done where you can modify, right, the polypeptide coding sequence of a protein which is typically not secreted, right, and make it secreted by adding one of these signal peptides. Sometimes that works. Sometimes it just screws things up. But it has been shown that that can be sufficient for secretion. Yeah. So that's the beginning of the process of targeting proteins to different subcellular spaces. And again, I would say that another important point here is that it's not like you're waiting for translation to finish, right, for this process to occur, but rather it occurs co-translationally. And from a protein folding perspective, that makes some pretty good sense, because once a protein is folded, right, in a stable, let's say, globular fold, getting it through one of these channels, which requires unfolding of that protein, would be even more difficult. So doing it before it's ever folded to begin with sort of makes some sense, just from first principles. Okay? So then we can zoom out and take a look at what happens. So here you have the nucleus of a eukaryotic cell. You have this rough ER where you have basically all of these complexes, right, pumping polypeptide destined to be secreted. And then there's this whole trafficking pathway, which I'm not going to go into a lot of detail. Suffice it to say that essentially those proteins, depending on their modification state, their association with other trafficking cofactors, can find their way into different pathways. Okay? One point that I will make is that during this entire process, there are often extensive post-translational modifications, right? And one of them that happens to be particularly true for secreted and receptor proteins is a post-translational modification called glycosylation, which is the covalent coupling of sugar groups to proteins to make so-called glycoproteins. Glycosylation. So glycosylation of receptor proteins on a surface of a cell are very important marks for mediating the function of those receptor proteins to interact with their appropriate binding ligands. But for our purposes, I just want to sort of cruise through here that there are different fates, right, that a secreted or a membrane protein might take. And it's interesting to think about like the sorting pathway where it goes through this interesting organelle called the Golgi complex or the Golgi apparatus. And then some of these are destined for something called the lysosome, which is a degradative organelle, or some of them are destined for so-called exocytosis, where they're secreted, for example, neurotransmitters. And then some of them are transported in this way, where they're displayed on the membrane and then of a vesicle, being shown here, a transport vesicle. And then when that vesicle fuses, this is kind of interesting. If you were to think about where the proteins are, they're sort of embedded in the membrane on the interior of the vesicle here. And then upon fusion of that transport vesicle with the plasma membrane, then the result of that is that those proteins can then be exposed on the outer surface. Okay? The take home here is that there are a lot of different trafficking pathways that all start, if you're not a cytosolic protein, start with engaging the endoplasmic reticulum and then being shunted into one of these pathways. Okay? For a host of other, in talking about, and let me just stress the point that this is particularly important for secreted proteins, proteins that are released from the cell, as I mentioned, and also membrane proteins, proteins that reside within the membrane in order to fulfill their cellular functions. Okay? Now, for a whole other class of proteins, right, that are made in the cytosol by the ribosome, don't have signal peptides for the ER, right, so they're not going to be secreted or embedded in the membrane. Some of those can just exist in the cytosol and function there, right? But many of them have to get into the nucleus, right, to mediate essentially all of these important functions and processes that we've been discussing in the class, right? So we can think, for example, of transcription. Is probably a good example. Transcriptional machinery, all those proteins that we talked about, transcription factors, RNA polymerases, sigma, well, I guess in this case not sigma because we're talking about eukaryotic cells, but other transcription factors, those are all proteins made in the cytosol, right, that need to get back into the nucleus to do their thing, right? And so you have this whole additional transport mechanism or pathway, which we refer to as nuclear transport. And this is a neat cross-section EM image of the nuclear envelope. Okay, so that's the surface of the nucleus. And what you can see is there are these circular structures that can be seen. And these are the so-called nuclear pores. Okay? So, but as I've mentioned to you earlier, nuclear pores are not just sort of tunnels for any protein that wants to go in or out of the nucleus to traverse the nuclear membrane, but rather they are gatekeepers for who gets in and who gets out. Okay? So if we look, this is again yet another complex pathway, but let me walk you through it. There's a theme here, right? Again, that I'll point out. So if we focus here on this image where we have the nuclear envelope, now we're looking at it from a side view, the nuclear pore complexes, which are shown here, those were those circular things in the EM image. So as you have a protein which is made in the cytosol but destined to make it into the nucleus for its function, it also has a consensus sequence, not called a signal peptide, but in this case called an NLS, which stands for nuclear localization signal. Okay, the NLS. So a protein bearing this NLS, as you might expect, will bind to factors. In this case, for the nuclear import complex, there are these two important factors. In fact, literally, their name is importin, not important, but they're import factors. Okay, so you have important alpha and beta. Okay, just to name the machinery here a little bit. And that alpha-beta heterodimer, those importin, right, which happen to be important, right, proteins will bind to the NLS and help target through the NLS important alpha-beta interaction the import, as its name would suggest, of that cargo, right, into the nucleus. And then, of course, you have this bound cargo. There's a series of steps that there's a lot of stuff going on here that I don't want you to get too bogged down in. But essentially, all of these extra steps are there to achieve two things. One is to actually deposit the cargo, which is actually down here, right? So you need to actually deliver the cargo, right, to do its function inside the nucleus, right? So that might be a transcription factor or something else that has to come into the nucleus to do its function. And so you have these other factors that are involved in displacing those important alpha-beta complexes from the protein that has been transported into the nucleus to release it into the inner space of the nucleus. And then you have a second set of processes, which I don't want, again, to go over in too much detail because I know we're running out of time. But the main focus here is making sure that those important alpha-beta proteins are recycled back out into the cytosol so that they can help other proteins get back in. Okay? So you need to, so all of this stuff here is basically critical for recycling the alpha-beta important proteins back to the cytosol, right? So they can be used again. Okay? So that's a little bit on the nuclear trafficking regulation, right? And you notice that there's a theme. So to get into the ER, there's a signal called the signal peptide. To get into the nucleus, there's another peptide called the nuclear localization signal. And it's actually sufficient for the NLS. It's a quite strong signal. So you can take like a fluorescence protein, like green fluorescence protein, and just tag it with an NLS, and it will light up the nucleus. Okay? So the pore complex is looking for that NLS, essentially, and that is sufficient to get cargo into the nucleus. And then the very last slide that I'll end with in the last minute, I'm sorry, I did not go short, shockingly enough. But this is the last slide, which talks about protein turnover or degradation. So proteins, when they're made, part of their natural let's say, protein circle of life, maybe, is that at some point they're degraded, right? Sometimes that is just part of the natural lifetime of a protein. So proteins can exist from anywhere from seconds to minutes all the way to hours, to some proteins that can last many days, right? That's more rare. More typically, minutes to hours are the lifetimes of proteins. And they get turned over. Alternatively, some proteins, as we've discussed, might be misfolded, might get damaged through oxidative damage or other types of insults. And those proteins need to be cleared out, right, as a quality control measure to make sure that they don't induce dysfunction. And there are many pathways for protein turnover, but the most, quote unquote, ubiquitous, so